Hello. Is it audible? Hello.
Hello, sir.
Hello. Is it audible?
Yes, sir.
Okay. Thank you. So, okay. So today we discuss the Lycopodium. Yesterday we discussed the general characters and classification of Pteridophyta, and another one is there. Then is the life history of the Lycopodium. So life history of Rainia. Yesterday we discussed. Is okay? Yes. Hello. Is it audible? Hello.
Yeah.
Okay. So today we discuss the Lycopodium. Here, the Lycopodium belongs to the division is the Lycophyta, and the order is the class is the Eugulopsida. Eugulopsida, and order is the Lycopodius. The family is the Lycopodiaceae family. The genus is the Lycopodium. Okay. So here the class is the Ligulopsida and Lycopodiaceae, and the Lycopodium. So the genus Lycopodium is around the 200 species are there. Out of the 200 species, only the 33 species are identified in India. So these Lycopodium we call commonly club masters. Club masters we are called. So the habit, this is the conspicuous plant of the Lycopodium grows in nature as a sporophyte. Okay, the structure of the sporophyte varies widely within the species are there. The plant are the slender, delicate, and baked stem is there. These species without any rhizome. Okay, that will be and Selago and Lycopodium, Fliparia are the species are there. What is the external features? External features. The plant body is distinguished into stem, root, and leaves are there. Okay, the plant body is identified. The stem, root, and leaf. The stem are either the pendant and branched or unbranched. In the Lycopodium, first from the root is a short-lived structure. Root system is the advantageous type of the root system and dichotomously branched. The leaves of Lycopodium very small, simple, and microphyllous. Very small in structure. The leaf gap is not formed. So the leaves are sessile and have the broad leaf base is there. Sessile means there is no petiole. The leaf is directly attached to the stem. They have unbranched and midbeams are there. The shape of leaves are very widely. Okay, these leaves are the needle-like obligate or oval structure is there. So in the Lycopodium, a ligule is not present. Ligule means a small leaf-like structure below the leaf. That is a small. That one is the ligule. Okay. Next one is the internal structure. What is the internal structure here? The stem. We can see the stem structure. Okay. So here we see the anatomy of PS of the stem. Here, this is the stem of the anatomy. We have in the practical section also this Lycopodium stem, transverse section of the stem. PS of the stem. You will do the in the practical session, and some slides also there. The stem section, which is a circular or even the outline or reveals three regions are there. What is the three regions? Here, epidermis, cortex, and stele. Any root and stem, which is differentiated into epidermis, cortex, and centrally stele is there. Similarly, here also Lycopodium is having the stem, the transverse section of the stem here, epidermis, cortex, and stele. So the epidermis is a single layer thickness is there. The outer wall of the epidermis are cutinized. The epidermis is a consist of the single layer, which is a protective layer. Upon the epidermis, a layer is there, that is the cutinized layer. The stomata is also present. The cortex is below the epidermis. Cortex is present, which is the several layer and broad. The thickness of cortex varies within the species to species. It may be several broader than the stele of the same size of the stele. So, in the homogeneous or heterogeneous zones are there in the form of the cortex consist of exclusively thin-walled parenchymatous cells or thick-walled sclerotia are present in this one. The middle zone is found with a thin-walled parenchymatous cell, and the innermost layer of the cortex, which is differentiated in the endodermis. Innermost layer is the endodermis. This one is the endodermis. We can see the clearly. Epidermis, cortex, and inner one is the endodermis. Very thin layers are present. Some here Casparian strips is also present. Okay, Casparian strips is also present. So in the endodermis, below in the endodermis is the stele is there, which is consists to 3 to 6 layer thin wall and rectangular cells are periphery, forming the pericycle is present. The stele is outer one is the pericycle, and innermost layer of the cortex is the endodermis. So the inner pericycle still consists of a solid core of xylem, the center surrounded by the ring of the phloem. So here the xylem is exarch and without any pith in the center of the stem. So here some members are asking page number here 57. Okay. So the Lycopodium, which is described as the stele, is the protostele. There is no pith, only the xylem is there. The xylem is surrounded by the phloem. So this type of the stele is being called as a protostele. So in the structure of the Lycopodium, varies these protostele in the different types are there. See here, though based on the stele arrangements here, the stele is the different types of there. Okay, means plectostele, actinostele, and microstele like that. Okay. So here, haplostele, actinostele, pectostele, and mixed plectosteles are there based on the species types. In haplostele, protostele, xylem is in the form of the solid, smooth, slender, and enclosed by a layer of the phloem. That is the uh, haplontic stele. So here, pith is not present. And next one is the actinostele. So the xylem is from the radiating ribs. Xylem rays are surrounded by the phloem. So that is the actin means rays. Actin-like structures are there. That is actinostelic protostele. So in the Lycopodium, planaria, xylem is rays and many as a 10 and are pointed. So here the actin is very advanced protostele than compared to remaining protostele, aploxtele, and plectostele. So in the plectostele, the xylem is consists which is divided into several plates like arrangements are there. So these parallel plates are to each other with the phloem occurring between these plates. That is the plectostele. And next one is the mixed stele. So mixed protostele, that one we call as a mixed protostele means the xylem is breaks into small pieces like that spherical in structure during the the total is spread into the stele. So this xylem is surrounded by the phloem. The phloem is um, totally occupied. This is the mixed protostele. Okay. Okay. This is the internal structure of the Lycopodium. This is a compulsory asking question. Internal structure of the stem of the Lycopodium. Here slides also there and the practical section also there. We can do the in practical session. Okay. The next one is the reproduction system. Root structure is also there, but it is not important. Leaf structure is not. So next one is the reproductive structure. The sporophyte of the Lycopodium is reproduced by the vegetative and asexual method. It is reproduced more frequently by the vegetative propagation. So here the vegetative reproduction means different types of the progressive growth and death of the older parts of the rhizome and the perennial rhizome, wherein the entire plant dies except the rhizome by the gemma cups or buds. Some budding characters are there. So root tubercle is also found in the cortical cells of the root as seen in the different species. So next one is the asexual reproduction. The sporophyte is the homosporous. Homosporous means all spores are in the same structure and same activities. That is homospore. Means morphologically and physiologically all spores are similar. That's why here homospore. Heterosporous means morphologically and physiologically is differ. That one we call as heterospore. So Lycopodium belongs to the homosporous sporophyte and produced by the formation of the spore. So here the sporangia are appear on the axillary leaves. That leaves we call as a sporophylls. Some the fertile leaves are there, some are the sterile leaves are there. Sterile leaves are they are not have these spores, sporangia. They are only possible in photosynthesis. But the apical region of these leaves, they are to form a club-like structure which are bear the sporangia. Within the sporangia, the spores are there. You can see the leaf, the plant body of the Lycopodium here in the apical region. So look at here, the apical region, there are the differ. This is the stobalus. This is the stobalus. Stobalus is nothing but cones. There are very different. Okay. Okay. This is the apical region. This one. The apical region here have the some a club-like structures are there. That's why the Lycopodium we call it the club masses. So these apical leaves are very different than the remaining uh, vegetable leaves. Means there are the sterile leaves. This one is the fertile leaves are there. Okay. So here this one is consist of the core. That one we call it the cones or stobal. So The sporophyll appear is different from the vegetative leaves are there. There are a different structure are there than compared to vegetative leaves. In certain species belong to the subgenus of the Eurostachy are there. So these the sporangia are a comparative large and kidney shaped structure. So between the sporophyll means these are the sporophylls are there. In the sporophyll, a sporangium is there. Kidney shaped sporangium, spherical shaped sporangium is there. Within the sporangium, the spores are there. At maturity, they are yellow in color, and each sporangium shows the multicellular stalk, and a capsule is there. The wall of the capsule is a three-layered thickness, and the innermost layer function as the tapetum. So, this is the development of the sporangium is the urosporangiate character is there. Okay. So here the sporogenous cells then undergoes to meiosis to form the haploid spores are there. The spores are separated and final stage is inside the sporangium. They are to the sporangium wall and liberated to the are adjacent to the spores by the wind. So each spore is a trilate mark is there. This is the spore. This is the spore of the Lycopodium. Okay, this one is a spore. This one is also spore. So these spores are applied very small in structure and tetrahedral and trilate mark is also there. So outer coat is very thick and shown the reticulate ridges like globe-like structure are there. So the innermost coat is very thin and uniform and granular. So a tridelic mark is there. Each Lycopodium spores have the a triradiate mark like this. Triradiate mark is there. Triangular radiate mark is there. Triradiate mark is there. This is the character of the Lycopodium. Okay. So next one is next is the gametophytic sexual reproduction. In the spore germinate to develop the gametophyte, which is known as a Protalis. That one there is the great diversity in the form the Protalis, which is also diversity from the species to species. So the spore germinate with a few days in the soil. The gametophyte are long and in diameter. There are subnate part in colorless and cylindrical, and the aerial part is green and lobed photosynthetic characters are there, which is colorless parts near the rhizome. Okay. After the second stage, what happened here? The prothalli are the yellowish brown and colorless without chlorophyll, or this saprophytic condition is there. Okay. So these prothallis are here 12 to 18 millimeter long, top or cone or plate shaped with a layer cylindrical and upper lobed part is there. So third one here, the Protalis is the colorless sporophytic. It grows and turns to the surface of the humus attached by the observed the hair projecting in all directions. It is regularly shaped with the tuberous and central body about 2 mm. The branch of unlimited growth are there. So this is the in the okay the prothallus develop. Next is the antheridium. The antheridium is either directly completely embedded in the slightly projecting part is there, which is very simple structure. Mature antheridiums consist of the body and single layer wall, includes the number of anthrozoids are there. Anthrozoids is nothing but spermatozoids. Okay. So here, these anthrozoids are liberated, means biflagellate anthrozoids are liberated and reach the oogonium, means archegonium cells are there. Archegonia sunken with their neck projecting from the gametophyte The neck of the archegonium consists of 4 vertical rows of each cell in 3 to 4 in height. So after the canal cells, neck canal cells are disintegrated to and fuse with the eggs. These protozoites directly fuse with the egg and to form the oospore. Then oospore again to develop into a embryo. So that embryo again to germinate and to form a complete uh, lycopodium plant body. This is the external morphology and the internal structure and the reproductive structure of the lycopodium. Is okay clear? Hello?
Yes, sir.
Okay. So in the exam point of view, the lycopodium means only the stem structure, internal structure of the stem. Okay. Transverse section of the stem is very important in the exam point of view. And the LS of the cone, strobalus, longitudinal section of the strobalus means reproductive structure. This is also very important. So strobalus wherein appear on the apical region of the stem, apical regions. Okay, they are to form these sporophylls are to form very compactly arranged to form a club-like structure, like this structure. Okay, so one specimen is also there in your practical session. So the plant of the sporophyte plant of the lycopodium means which is differentiation to root to stem and leaves. The very slender like structures are the very smooth in structure. This is the lycopodium. Okay, so here lycopodium is the steel is different types of steels are there. That is the plectosteel, actinosteel, mixed steel. Okay, lycopodium is protosteel condition is there. Within the protosteel again different species to species. This protosteel is there. Protosteel means there is no pith. Xylem is centrally arranged and the xylem surrounded by the phloem. So that one we call as a protosteel. So here protosteel again differentiated into different types are there based on the species. Actinosteel, plectosteel, and mixed protosteel like that. Okay, Nandi. So next one is the structure of the strobalus. Strobalus means cone, nothing but cone. Is bearing the sporophylls are there. Upon the sporophylls, a kidney shaped sporangia is present. Within the sporangia, here number of spores are there. This is see here, this one sporangia. This total we call as the strobalus or cone, nothing but. Okay, clear?
Yes, sir.
So next we are coming under to the equizitum. So here this equizitum belongs to the spinopsida member. Okay. So this equizitum belongs to the spinopsida and class is the Calamopsida. Order is the Equizitales, family is the Equizitaceae, and the genus is the Equizitum. Okay. So these plant bodies commonly we call as the horse tail. So we are calling here horse tails means this is looks like a horse tail like structure is there. See here, horse tail like structure is there. That's why this commonly we call as a horse tail. Okay, around the here 25 species are there in our India. The plant body is the, here also the sporophytic plant body is there. This is also very important in the examination point of view, means practical examinations also. One specimen is there. Okay, one specimen is there. Equivalent specimen is also there, which is also coming in the essay question also, theory also. The habitat. Habitat means the equivalent is very variety habitats are there. to different localities in the forest as well as the swamp areas, and in the dump localities, they are shaded areas they can grow. So what is habit here? The equisetum is the conspicuous plant in the sporophyte, which is the diploid. Sporophyte is nothing but diploid condition. So all the species are rhizomes and perennial hair. They are the slender and generally creepy. So the subterranean rhizome is much branched. Some of these branches grow erect and became aerial, which are usually annual herb-like structures. Are there. The plant body is very slender like structure. The diameter stem is only 0.5 centimeters to form the 2.5 centimeters in long. Here, external form. What is the external characters are there? The sporophyte equisetum is differentiated into leaf and stem and roots also, similarly like lycopodium. Okay, leaf, stem, and roots are there. So this is have the scale like leaves are at the each node. So some scale like leaves are there at present each node. The rhizome, the rhizomatous stem grows horizontally, sometimes is penetrating up to the depth of the 1 meter under the soil surface. So here there shows the nodes and internodes are present. The stem is shows the nodes and internodes. At each node of the whole of the scale like leaf are present. Some leaves are present. So here at the base of the each node, there are intercalary meristems are there, meristematic tissue that is responsible for the growth of the internodes. Sometimes is present because the stem is weak and easily gets pulled upon the internode. So here the number of ridges meaning on the leaves at the each silica. So they shows ridges and furrows are there. And the stem is the ridges are some number of ridges and furrows are there. The number of ridges is many as the leaves at the each node. So each ridge leads to the same leaves in the that one leaves on the each node. So aerial branches produced from the nodes and underground the rhizome, which is vertically above the ground. So they form green in color and chlorophyll. They can do the photosynthesis. So some the non-photosynthetic leaves also there, which are bear the chlorophyll, means sporangia or stromal. Okay. So the aerial, that is the sterile branches may unbranched or branched structure is there. These sterile shoots, much branch shows the branches are tertiary order in some equisetal species are there. What is the stem structure here? You can see the stem structure. Internal structure of the equisetum means this is the internal structure. What is the stem here? So, so the stem is the transverse section of the stem is a circular but wavy in outline due to the presence of ridges or furrows are there. Okay, some wavy nature is there. So based on the ridges and furrows are there, so the outline is looks like a wavy nature is there. The stem is differentiated into three regions are there: epidermis, cortex, and stele. So the epidermis consists of a single layer. This is outer layer is the epidermis, which is consist of a single layer, and the outer wall is the very thick layer is there, which is made up of the silica. Silica deposits are there. So, which is if we feel the stem of the equisetum, very tough and embedded on the protuberance and within the fine granules are like rosette and spines are there due to the silica deposits, which is very tough one, uh, which is very hard in structure. So because the deposition of silica, the surface is very rough. And the epidermis also provide a very thick cuticle is also. Each stomata, some stomata is also the stomata is having two pairs of cells are there. So the inner pair of the guard cells are completely over arced by the pair of the subsidiary cells are there. The stomata in the equisetum are unusual and characteristic because the stomata initially undergoes successive longitudinal division, the deposition of the silicous celsaceous brand-like thickness between the accessory and guard cells. So here cortex is very broad and highly differentiated into the function of photosynthesis and mechanical subgroup also. So which is differentiated into hypodermis, middle cortex, and endodermis. So hypodermis is the scleral chromatous tissue which gives the mechanical strength of the stem. Hypodermis. Below the epidermis is the hypodermis is there, which is made up of the scleral chromatous. Means very thick cell wall, cutinized cells are there, lignin cells are there. And below the hypodermis, parenchymal tissue is there, that is the middle cortex, nothing but. And inner one is the endodermis, lowermost layer of the cortex, we call as a endodermis. Okay. The vascular canals are present in the cortex regions also there. Some gaps are there, that one we call as a vascular canal. They are contain the air chamber or aeration. Due to the aeration, here air gaps are there. And here the steel is the siphonosteel is the means centrally when it is there, there is the siphonosteel is there. Okay. So here also cranial cavity is also there in the vascular condition. See here, cranial cavity is also there. The central one pith is there. And cranial canals also there in the vascular system, means metaxylem and protoxylem are there. Okay, this is the internal structure of the equisetum stem. Okay. So next one is the what is the reproductive structure of the equisetum. What is here reproduction structure? This is also very important in the exam point of view. The sporophyte is reproduced by the vegetative as well as the asexual reproduction, like the Lycopodium. Here also stomata is there. Okay, is it audible? My voice is audible.
Yes, sir.
Okay. So here, the vegetative reproduction means the vegetative propagation takes place by the tubers or some breakdown of the rhizome. This is the vegetative reproduction. In the asexual reproduction, so here asexual reproduction is the homosporous condition is the same, just like Lycopodium. Equisetum is the homosporous condition, means the all spores are alike, means same structure is there. These tubulars are solitary or terminal, means single and apical region of the shoot. There are some specialized fertile branches are there. That fertile branches bear these stabile. like this. So that's why, based on these stabile, they are looks like a horse tail like structure. That's why the ecosystem we call as horse tail. So each tuberous is a spindle-like structure and consists of central axis where the several mushroom-shaped sporangiospores are there. The central axis is there. So on the central axis, in the lateral side, some sporophylls are there. So upon the sporophylls, bear the sporangia. Okay. The sporangiospores are projected at the right angle to the central axis. Below all the sporangiospores are central bear the whole specialized leaves, so they are united to form a cup-like structure that is the annulus. So this is okay. The annulus is more conspicuous in the specialized subalus. So each sporophyll consists of a short cylindrical stalk with a distal and expand from the circular plot and plated disc. So the disc is right to the right angle to the stalk because the lateral presence of the adjacent the disc, and each disc contains the hexagonal outline, and the one side of the disc is 5 to 10 meters in the sporangia BS. So these compact arrangements of the peltate discs provide the protecting cover for the sporangia located under beneath. So the sporangia are born on the undersurface of the disc sporangiophore, projecting horizontally toward the central axis. There are the this is the sporangia. So they are the cylindrical and shark-like structure. It matched each sporangia consists of two layers wall within each spore are present. So here eusporangiate condition is. So after meiosis means sporogenous mother cells undergoes to meiosis to produce the haploid spores are. So some these after development of the spores are liberated and there are spherical and some uninucleate structure is there. They can so here four layers spores are there. So that is innermost layer is endospore and which consists of the cellulose, outer one is exospore, middle one. Like okay. After the development of these spores, their hygroscopic nature is there, and they are some spirally coiled around the spore under the moist condition. They are in the dry weather. They are uncoiled to present cerebral like strips are there. This one we call as the elater. So here the ecosystem is not a comparable to the elater of bryophyte, but they are to dehiscence. Main these elaters are dehiscence in the core. Okay, and hygroscopic movement of these elaters may assist in the dispersal of the spore. So this is the ecosystem. After the liberation of the what happened here, similarly, the spores will germinate and to produce the gametophytic plant. Okay. Hello. This is the ecosystem.
Yes, sir.
Okay. So here in the ecosystem, here the external morphology of the ecosystem, the plant body of the ecosystem, that is the specimens are there, which is also coming under the examination point. External morphology of the ecosystem and stem anatomy, transverse section of the stem of ecosystem, and another one is the stabalus, asexual reproduction of the ecosystem, stabalus structure of the ecosystem. This is also very important. Specimens also there, slides also there. The theory also is coming the Sobalis, cone of the equilateral. Describe the cone of the equilateral. They are asking like this question. Okay, clear? Okay.
Is
So the next one, another topic is there. That is the Marsilia. Marsilia, structure of the Marsilia means here. This one is the. This is the Marsilia plant. So this Marsilia is the sporocarp means sporophyte plant body. So this is the division is the Philicophyta, class is the Leptosporangiopsida, order is the Marcilius, the family is the Marciliaceae, genus is the Marsilia. So the external structure, the sporophyte, the Marcel plant body we call as the sporophyte, means a diploid condition is there. That's why the sporophyte. So these Marsilia sporophyte may be aquatic or amphibious in habit. Means they can grow on the in water as well as uh, in the wet places, wet areas, shallows area, ponds, near to the pond. Okay, so very mud area means terrestrial conditions are there, aquatic habitat as well as the terrestrial habitat. So these conspicuous plant of the Marsilia is a sporophyte, which is resembled to a fern-like structures are there, which is differentiated into stem, leaf, and root. See here, the root and stem means nothing but is modified as a rhizome. So there is no specific rhizome, but a leaf is there. Long petiolated leaf is present in the Marsilia. Stem, leaf, and root. So all the species stem are slender and branched and rhizome is there. The rhizomes are skipping with is undefined growth and were found in the young and just below the soil surface. Okay. There are the less frequently branched with either dichotomous or lateral branches. So here, the leaves, the rhizome here, what happened here? The lateral case branches are produced near to the or below the leaves, but they never from the axis of the leaves. So the rhizomes are shown in the distinct nodes and internodes also there. So next one is the leaf. Leaves are very here in the Marsilia leaves are very specialized. The compound leaves are the pinnately compound leaves are. Okay. So leaf here, long petioles. These are alternate and arranged in two rows and dorsal side of the rhizome. They are the petiolated and quadrifoliated. Quadrifoliated means four leaflets are also there. This is totally one leaf we call as. This total one leaf. Okay. Four leaflets, quadrifoliated means four leaflets are present. So that's when that character is we call as a quadrifoliated compound leaf. So the length of the petioles varies with the shallowness of the pond in which it is grows. Very long petiole is there. And pinnately compound leaf. This one we call as a pinnately compound leaf. The of the four leaflets are found in the distal pair which are slightly higher. Two forms are distal pair which is very height in higher in slightly height. So the single leaf base is divided into four leaflets. That is, there are the three successive dichotomy. And the veins are leaflets are dichotomously branched with the vein with the inner construct and the cross sections here. So the succinate venation is present means the younger leaves are they can spirally arranged. Their younger leaves are to carve-like structures are spirally arranged, spring-like structure, carve-like structure. That condition we call as a succinate venation. Succinate vernation is there. As the plant, they are start to produce a bodies, some a pear-like structures are there. That one we call as a sporocarp, small at the base of the petiole. At the base of the petiole, a pear-shaped structure is there. That one we call as a sporocarp. This sporocarp to produce the spores in the reproduction system. They are involved in the reproduction system. Okay. The next one is the rhizome transfer section. So here, rhizome transfer section of the Marsilia. This is also in the outline, which is circular structure is there. This is also differentiated into three regions: are there epidermis, cortex, and stele. So epidermis is a single layer without any stomata. Means they are submerged into the soil. So that's why there is no stomata. The cortex, which is very differentiated to outer cortex and middle and inner cortex. So the next epidermis is to that is the outer cortex, which is the parenchymatous tissue, and many air gaps also there, which is the different divided into air filament like structures, trabeculae. That one we call as a trabeculae. Some air chambers are also there. So due to the air chambers, this is a specific character of the aquatic plants. Air gaps are also there. So they store the air. So this is the very aquatic character. So in the inner cortex is solid consists of a ring like a wall is there. This is below the air chambers. There are to give the some strengthen to the rhizome. Some sclerenchymatous tissue is there. That one is the innermost inner cortex. Okay. So another one here, the stele is the very differentiated type of the stele is there. That one is the amphifloic cyclostele. Centrally, pith is there, so that's why here pith is present. That is called as a cyclostele. But amphifloic cyclostele is present in the Marsilia rhizome. Amphifloic means amphi ante both sides. Amphi means both sides. So here, the outermost layer of the stele is pericycle with a single layer rectangular. Amphifloic cyphalosteles means the xylem is surrounded by the both sides of the phloem. The xylem is surrounded in inner side as well as outer side by the phloem. That one is the amphifloic cyphonostele. Means inner pericycle is there, inner endodermis is there, and outer pericycle is there, outer endodermis is there, and outer phloem is also present, inner phloem is also present. So in both sides, the phloem and endodermis and pericycle is present. So this is a specific character of the Marsilia rhizome, amphifloic cyphonostele. Amphifloic cyphnosteles means the xylem is surrounded by the both side, means inner side and outer side of the xylem. The phloem is present. That one character is amphifloic cyphnostele. Is okay. So next one is the petiole structure. So here petiole is also there in your practical session. The petiole is there. This is also coming on the theory also. So petiole is also a circular in the outline as the stem which is consist on the endodermis of the single layer and rectangular cells. So here the petiole is also having the number of air chambers also. So below the epidermis is the hypodermis is there, that is the sclerenchymatous tissue. The hypodermis is to give the strength of the petiole. The inner to the hypodermis is the middle cortex, have the ring of the air chambers that are separated by the single layer septa. That one is septa is nothing but trabeculae. So the inner cortex is compactly arranged, which is made up of the parenchymatous tissue. So innermost layer is the endodermis is there, which is the rectangular in outline. So the central even steel is there. The xylem is located in the middle, V-shaped with the two arms of V-shaped each other. So the wide opening V is towards the axial side, and the middle each stand to the two larger tracheids that form the adaxial side of the petiole. Okay, in the middle of the each stand one two largest tracheids to form the metaxylem are located towards the either, and the smaller tracheids form the protoxylem, which is situated making the xylem exarch. The xylem is surrounded by the soil. So this is the petiole. Petiole is also there in your section cutting. Practical. And the next one is the reproduction. Next one, reproduction means here the marsilia is reproduced by the sporocarp. Marsilia, their reproduction means they are the vegetative and asexual reproductions are there. So in the vegetative reproduction means by takes place by the means tubers and the rhizome will break down into small pieces. But in the asexual reproduction, there is a special type of the sporocarp is there. This marsilia is the heterosporous type of the sporocarp is there. Heterosporous means two types of the spores are there. Two types of the spores are there. Microspores and megaspores. Heterospores. Okay. So in the sporocarp, the microsporangia they produce the microspores and megasporangia to produce the megaspores. Inside the sporocarp, already these sporocarp appear on the base of the petiole. A pear-shaped structure is there. Base of the petiole. Here, a small pear-shaped structure sporocarp is there, brown in color. Inside the sporocarp, two types of the sporangias are there: microsporangia and megasporangia. Microsporangia to produce the microspores and megasporangia to produce the megaspores. Okay, so in the sporocarp are born in the lateral petiole. They are attached to the means of stalk, which is often called a small pedicel is there. The number of mode of the attachment of sporocarp is very species to species. So here they are to when the bifacial sporocarp is split to open along the marginal and dorsal anterior, the ventral side, it is open towards the two half. It will be opened into two half. Okay. So the sporocarp is gelatinous color. The sporophore is present to the posterior side. It is extended to the dorsal and anterior side, and ends somewhere the middle of the central core is there. In the inner side here, two rows of the microsporangias are there. A small stalk is there. In the small stalk, the higher side, two microsporangias are there. And the middle one is the megasporangium, one megasporangium. So this covered a inducium, which is covered a inducium is there. Okay. So this is the sporocarp. Okay. Okay, clear? Huh? Is anybody? Okay, clear? Hello? Hello? Okay, clear? Anybody?
Yes, sir.
Okay.
Yes, sir. Yes, sir.
Okay, thank you. This is the Marsilia, the external morphology. They are asking the Marsilia external morphology and anatomy of rhizome. Anatomy and rhizome means the transfer section, PS of rhizome of the Marsilia, that is also important. And another one is the petiole, anatomy of petiole, transfer section of the petiole, that one is also important. And the sporocarp, Marsilia sporocarp, structure of the transfer section, and VLS, RLS structures of the sporocarp. This is also asking the exam point of view. Okay. Okay, you please you note down these questions also. What is the important topic in your exam point of view? This is also important. Okay, Nandi. Anybody?
Sir.
Is okay?
Yes,
So
sir.
tomorrow we discuss the stela resolution. So in Lycopodium here, Lycopodium, Equisetum, and Marsilia. These
Yes.
three topics are very important in your theory as well as the practical also.
Okay, sir.
Okay. In the Lycopodium, specimens also there. External morphology and transfer section of the stem. The stem is here, Lycopodium, the protostele. But Marsilia Lycopodium stem is, what is the stele character? Amphifluic siphonostele. Marsilia rhizome stele is the amphifluic siphonostele. Amphifluic siphonostele means the both side of the phloem is present. Xylem is surrounded by the both sides, inner side as well as the outer side. Outer side. So that is the amphifluic. Amphi means both two. Floic means phloem. Xylem is surrounded by the both sides, inner side and outer side. The phloem is present. That is the special character of the Marsilia rhizome. Okay, Marsilia rhizome is the special character. The amphifluic siphonostele.
Okay, sir.
The Marsilia petiole is also there. Petiole is also important, and the sporocarp is also important. Equisetum, the morphology, external morphology of the equisetum and stem, transfer section of the stem, and strobalus, LS of the strobalus, longitudinal section of the strobalus. This is also important. Okay.
Okay. Okay, sir.
Okay, sir.
Okay. Okay. So be present. Some more people. What happened here? Some members are not attending the class.
They are in work. They are in work, sir. In their works.
Their work also. This is also important for your
Yes,
degree.
sir. Yes, sir.
Okay. So thank you. Just to intimate your friends. So thank you for attending your class.
Okay.
Thank you, and thank you everybody.
Okay, sir.
Thank you, sir.